Okay, so let's take a look at this effector phase. We're back looking at CMI, and let's take a look at this effector phase. So here's a picture in which you have a target cell. Here's a CTL, and these cells are not round actually. What they will they what they it's referred to as become polarized, in which you'll see a leading edge which appears to be sensing out for things, and then a lagging edge, and so they become kind of elongated or depolarized. They seek out interact with that surface, form a synapse. They rearrange, and which leads to then the dumping out of granules, and then this leads to then destruction of the cell. We can see that in terms of the granules. We can see, for example, here's a T cell interacting now with a target cell. The granules have been dyed red. A few minutes later, what you see is these granules are moving from the back of the cell to the front of the cell in that area. Well, here's a start. Within 1 minute, you can see the granules moving across the cell to they interact 4 minutes later with that surface. And then after 40 minutes, you can see the cells have started to undergo digestion or apoptosis and destruction in the presence of those enzymes. Now, one of the key things that's important is the formation of these pores from these porphyrin monomers, and they're sort of chemically kept as monomers within these granules. And when they're dumped, there's a sort of a pH change, at least, that occurs that causes these monomers of porphyrin to form into a pore. Now, we've already seen how pores are very important for complement, and now we see for the CMI they're important here as well because they get formed in the host cells, and they lead to the destruction. The loss of the integrity of the membrane occurs. Water flows in and out. ATP flows out. Calcium flows out. All those things, which are extremely bad for the target cell. You can see here from this electron micrograph, actually, you can see the perforins in a sort of where the membrane's been lifted off, and you can see them. They're formed these neat little basically nanorings across the cell membrane. Now, what I'd like to do now is sort of relate this to the research I'm doing to show you kind of a real world way that the research that I'm doing in my lab, we're doing in our lab, that's one of the projects that we're working on, has to relate of using cells, engineering cells, for to take advantage of their ability, T cells, their ability to kill tumor cells. And what we've done is we can take a lipid chemical dimerizer, mix it with a protein scaffold that we've assembled that has what this looks familiar, a single chain antibody on it. In other words, a variable region that's been recombinantly fused to this molecule. When we put those together, they form rings that have phospholipids on them, and those phospholipids enable that nanoring to then insert into the lipid membrane of the T cell membrane, which means that the T cell is now functionalized. We've artificially functionalized it so that it will target what we want. And in our case, we'd like to target cancer cells through their specific antigens. And in this case, what that means is that we're looking for is that these functionalized T cells are now going to go after the cancer cells and individually start killing these cancer cells. The CTLs keep doing this, in this case, totally artificially. Excuse me. So now let's look at some cells in this experiment. This is live cell imaging. which are going to take our nano functionalized cells. In this case, there are no functionalized cells here. And if we look at this, what we see is these big cells that are red, that have been labeled red, are glioma cells, brain cancer cells. They're really big. Notice, for instance, all these cells move around a lot. Yeah, that's right. They move all over the place to have these long tentacles that are forming. This cell right here is dividing. Did you just see that? This brain cancer cell just divided. And now this group of cells up here that's not um, labeled are breast cancer cells. So there's two different types of cells that are here: brain cancer cells and breast cancer cells. And what you find is that, and then these little wiggly guys are our T cells. They're moving around and they're interacting with with these cells, um, touching them, finding out if they have them, if they can interact with them. But they don't really do anything. They don't really do too much with these to these cells. Now, if we take instead, we take our cells, and now what we're going to do is we're going to, let me see if I can, okay. We're going to take, again, to remind you, these big cells. These big cells are these red cells are the glioma cells. These cells here that are kind of grouped together uh, are um, um, are the breast cancer cells. And then you'll notice there's some cells, like these cells, are starting to die. Now if we look down here at this bottom here, you'll see these cells here, and you notice there's lots of T cells around it. They're swarming over it, and the cells are dying like crazy. Now look at this cell up here. This cell, this lone cell, is dividing into two. Two T cells just recognized it. Another T cell just recognized it, formed the two daughter cells, and boom, it's dead. It collapses into a mass. And notice the membrane. This membrane is frothy, and that's and turning inside out. That's a characteristic of apoptosis. The chromatin starts to light up. It becomes very, you can see the fluorescence of it. And then what I love, my best part of this, is they blow up. They just blow up. It's so great. And I could watch that all night long. And then all these breast cancer cells are killed once we functionalize them. What's really cool too is that the cells, the brain cancer cells, are not touched, and so they don't have the antigen that's on them. Only the breast cancer cells have it, and so the T cells that have been functionalized will only go after and kill those breast cancer cells. So I sort of forgot to mention that the antibody that we put on those cells, the T cells, is specific for those breast cancer cells and not for the glioma cells. So we're currently looking at using this as a potential new therapy, a new way of engineering cells for immunotherapy. And it's been a lot of fun, and we're making a lot of progress in designing these in different ways. Excuse me. Now, again, this is just kind of looking at, with some pictures at what a cell looks like when it goes under apoptosis. This is a healthy cell. Here's the chromatin. You can see the chromatin in here. It's sort of the nucleus. And you can sort of notice that the nucleus is really large relative to the rest of this. In some cells, that's true. There's just a lot of chromatin in the plasma. The actual cytoplasm seems to be a minor part, but everything goes on in here. As the cells become necrotic, they form bubbles and stuff, and their membranes start to become destroyed, and their chromatin becomes cut up into pieces. There's DNases that go in and start to cut up um, the chromosomes. And then if you look at late stage, basically the membrane is totally fragmented, totally gone. The nucleus is just a little nub that's being destroyed, and essentially the cell is dead. And you saw that. You can go back and look at that movie, and you'll of some cells we've run in the laboratory, and you can see that. So kind of as a response, let's take a look at all these T cells that we've so far looked at. We've looked at, if we look at over here on the right, we've looked at Th2 cells, and these Th2 cells are really involved in, you can see they're involved in bacterial toxins, killing bacterial cells, responding to responding to B cells to activate and produce antibody and plasmacytes, and these antibodies have strong antitoxin potential. Th1 cells, which are involved in interacting with antigen processing cells that interact with MHC2 as well. Oh, jeez. Sorry about that. Th1 cells, which are involved in interacting with antigen processing cells and activation then of what cells producing IL-2. That's important for the activation and of cytotoxic T cells, pre-TK cells, pre-T killer cells that then go in and will destroy virally infected, potentially cancer, anything that's infected in which you have internal antigens within the cell, such as a virus, and they interact and destroy the cells. Of these types of T cells, only the cytotoxic T lymphocytes, the T killer cells, actually are involved in direct cell killing. Th2 cells are involved in helping, and so, hence their name, helping production of the antibody. Th1 cells are basically help to generate the CTL's response. So if we put this all together, I know this is frightening. It looks pretty frightening, but you've seen basically this before. And this part here is all the part that's involved in antibody production. And so this is annoying. And then remember, here's the Th1 cells, and this part here is involved in the CMI, or affection for intracellular pathogens. So this is AMI over here. Sorry, this is hard to write here. There we go. And CMI is over here. And there is an intersection. That intersection between them, that intersection is really at these Th1 cells. So this intersection with Th0, Th1. So if a macrophage takes up, for example, a virus or dendritic cell, can go and this can lead to the production of Th1 cells, which then can go interact again with the antigen processing cell and lead on to a CMI response and CTLs. And at the same time, what could happen is if there's IL-4 around, you could have Th2 cells produced, and which leads to antibody production. And so that's, you see commonly, what we'll see commonly is that, for example, for a viral infection, you won't have a strict CMI response or for and a strict AMI response, but you'll have a cell-based immunity response as well as antibodies being produced. Whereas for bacterial antigens, for bacteria, because you can't have a CMI response, because unless the bacterium can reproduce within the cell, you can't have that, then it's pretty strictly on the AMI side. Your AMI side, which is produces antibodies. So we're going to end here. And this will end our discussion on this lecture video on the CMI response. And I will see you in class.